DNA transcription. The key concept here is that transcription converts a gene into a single-stranded RNA molecule. So the product of DNA transcription is a single-stranded RNA molecule. All right, so here we see in this image an outline of a eukaryotic cell. A eukaryotic cell has a nuclear membrane. What I want to make very clear here is that the DNA is never going to leave the nucleus. It has to stay in there because it is safely kept inside the nucleus. This is why RNA is needed, because RNA codes for that DNA message, only in a different language, if you will, the RNA language. That RNA is going to travel outside of the nucleus into the cytoplasm, and then it's going to be converted from an RNA into a protein. The central dogma is an umbrella term used to denote all of the processes that encompass the transformation of DNA into RNA and then into a protein. So those three processes are what the main topic of this unit are. Replication, which we've gone over in the previous lecture. This lecture's topic, DNA transcription, and then next lesson's topic, DNA translation. And then along with every process, we are going to start with a reactant, so ingredients, whatever we start with, and then we're going to have a product. So in replication, we start with DNA, and then we form 2 molecules of DNA. In transcription, we will start with DNA, and we will end with RNA. And finally, in translation, we will start with RNA, and we will end with a protein. All right, but before we go over the specific steps of transcription, we must go over the differences between RNA and DNA. So RNA has 3 very important differences with DNA. RNA does not have a deoxyribose sugar. RNA has a ribose sugar. It is structurally different from a deoxyribose sugar. The second difference is that RNA has uracil, represented here by a U. RNA uses uracil in place of thymine. That means that uracil is going to be complementary and it's going to pair with adenine. The last difference between RNA and DNA is that RNA is a single-stranded structure. So if you'll remember 2 lessons behind, we went over the structure of DNA and how it was a double helix with uniform width. RNA has a different structure altogether. It is a single-stranded molecule. All right, a quick overview on transcription before we dive in. So transcription is going to start off with the DNA molecule. It's going to unwind a gene, like we can see here, where the nucleotides are present. It's going to break up that DNA molecule, and it's going to start coding for an RNA message. The RNA here is depicted in red. The DNA is in blue. Once that message has been completed, the RNA will separate from the DNA, and that will be the product of transcription. Much like replication, DNA replication, transcription uses enzymes. So the main enzyme that's used during DNA transcription is RNA polymerase. Remember DNA polymerase? Well, RNA polymerase is the main enzyme for transcription. So that DNA polymerase is denoted or represented in this image as a big yellow blob. And it's going to form what we like to call a transcription complex. So that's just a group of proteins that are going to gather around the DNA molecule and assist with the process of DNA transcription. This transcription complex is going to recognize where gene starts. So here you can see in that image it's represented by a start site. And at that start site, that is where the DNA molecule is going to unwind. And the RNA polymerase is going to slide along that DNA molecule until it reaches the end of that gene. Right? But not only that, that RNA polymerase is going to start building that RNA message. So that RNA message is represented here in red. The DNA is in blue, just like the sketch. So as I said, the RNA polymerase is going to slide along the DNA molecule until it finishes the DNA message, the gene. Once that gene has been transcribed into RNA, that complex is going to fall apart. You can see that by all the segmented yellow blobs that have now fallen off of the DNA molecule. And what's going to happen next is the DNA molecule is going to wind back up, and the RNA message is going to detach from the DNA molecule. Okay, so the product of transcription really are 3 different types of RNA. The first one is mRNA, messenger RNA. This is the message that leaves the nucleus. This is going to be the part that gets turned into a protein. The second part, rRNA, is ribosomal RNA. This is going to help that messenger RNA become a protein by forming part of the next complex needed for translation. And finally, tRNA, transfer RNA. This is going to also help out in the translation complex by bringing amino acids from the cytoplasm to a ribosome. All right, you can see a lot of similarities between replication and transcription. Transcription and replication both involve protein complexes, also called enzymes, that help and assist with the base pairing. These 2 processes have different end results, however. Replication copies the DNA. Transcription copies a gene.